Hello, everybody. I am going to record this class. It's on the, this will be Tuesday, December 2nd, and it's the class on the fish, on the basically starting with fish. You watched, by now you should watch the, the videos on the history of the earth, and you should then the quiz today. Um, so this is the beginning. So actually, I'm sorry, December 1st. This is the beginning of the actual class on fish. Let me share screens. So we are in week 2, and I have 3 PowerPoints for you. Hold on, share screens. Okay, so these are the 3 PowerPoints, and I'm going to go through them in order. So I'm going to start with lecture 1, lecture 2, lecture 3. I actually might not finish lecture 3 today and then do it tomorrow. I'm actually recording this on Wednesday, so that's why I said December 2nd. So if you open up the PowerPoint and take notes, I'm going to go through the beginning. Oops. And sharing the PowerPoint. Okay. I'm going to be, the reason I'm showing the PowerPoint this way is that I'm going to be writing things here as well as using the camera. So if there's a particular word or something that you might not be familiar with, some concept, I'm going to explain it here. So first, I know we have some new students. We have at least 3 new students. What you are catching up to the rest. I had asked previously if students had the genetics class or any type of genetics class, or if they were familiar with Hox genes. And a couple were not. So if you are not, Hox genes are basically supervisor genes. They turn other genes on and off, and the reason I brought them up is because it's believed that the reason we have such diversity of animals in the planet has to do with the duplication of these sets of genes. For example, in invertebrates, they have only one set. Okay, this is one set, not one gene. One set of these supervisor genes. And the amazing thing about these genes is that the actual DNA code is the same, whether if it's a butterfly, a monkey, or a person. So these control genes is something that has been conserved through evolution. Now, in invertebrates, they have one, and then in vertebrates, but without a jaw, having a jaw is a major event, I should say, in evolution of vertebrates. So these jawless vertebrates have 2 sets. So there's a replication or duplication from the invertebrate. And then when we get to jawed vertebrates, we have another duplication of the second duplication. So now we have 4 sets of these genes. And it's believed that this duplication allowed for many phenotypes, or for these extra genes to go and explore new shapes, develop into new forms, and see which, what would be adapted, what would not. Okay. So again, Hox genes influence many other genes. We call them a supervisor gene. They basically turn genes on and off. One example this is when they were discovered. On the left, you have a normal fly with little antenna. If you take the Hox genes for those antenna and replace them with the Hox genes that control when a leg and where a leg is made, and you put it there, these flies end up growing legs in their heads. These legs don't work, because they don't have the same connection to the rest of the body like they do where they belong, but they form. Another example here, and this was so the same type of experiment. You take a Hox gene that controls something, and you put it out of place and see what it does. Okay, in this case was eyes, and you took these genes, put them somewhere else, and this is in flies, and they grew eyes where they didn't belong. Okay, and if you want to read up on this, this is the reference right here, Science in 95. Okay, so the reason I am bringing this up is again to know that the this duplication of these Hox genes is connected to the history of the vertebrates and the evolution of these different new new shapes and forms. So let's start with ichthyology, or the fish. So first you saw the videos, and you remember that when we look at the evolution of life on Earth, we get to the Cambrian, and that's when we have this big explosion of fossils, the Cambrian explosion. Then all the life was on Earth. I mean, was in the ocean. There was nothing on land. So it's fish that we're going to be looking at. And these are the basic fossils of these ancient fish. The superclass is called Agnatha. Okay, and this means jawless fish. So right up here. Okay. So basically, when you have the A in front of a word, it means without. This is in biology. So without whatever is after, and nathos or natha abbreviated, it means jaw. Okay. So these particular fish, there were three big groups. And again, don't worry so much about the actual taxonomy. We have the ostracoderms, cyclostomes, and the conodonts. Okay. The ostracoderms are going to be broken down to the heterostracans, excuse me. Then examples, the Tetrapedus morphi and the anaspids. And then the other group is Cephalopedus morphi. Yeah. The cyclostomes, we still have living organisms of this. There are two big groups, the Myceni and the Petromyzontidae. And then the conodonts are this strange group that went extinct. And we just have really scattered fossils of what they were. Okay, so here's where we are. So we are past the Cambrian, but not too far. And you see that we're going to cover the Ordovician, Silurian, Devonian, Carboniferous, Permian, all the way into the Paleozoic, all the way to Mesozoic. And these two groups, hackfish and the lamprey, which are the Myceni and Petromyzontidae, are the only ones that are survive until present times. See, the conodonts were this big and really common group, and we don't know much about them. Again, we don't have a lot of fossils. And if you look at the Devonian, it's called the age of fish because you see everything. Just about everything that fish thing that lived was present in the Devonian. So not a good time to go snorkeling. Okay. So again, the fossils of these chordates are scarce because again, when we went through the last class, now they have to fossilize under the right conditions, and the conditions are not there. You don't get a fossil, so we don't know they existed. Okay. But there are a few, including this one called Picaya from the Burgess Shale. Burgess Shale is that famous fossil bed in Canada. Now this appears to be, let me show you a picture so you know what I'm talking about. This is Picaya. Appears to be a cephalochordate because it does have that notochord and the body with segments. So it's an idea that this vertebrate plan or the vertebrate evolution was occurring at that time. Another record we have is Haikuela. Now this is from the early Cambrian, so again really ancient. Was found in China, and these just about 3 centimeters in length, so just over an inch. And again, they were fish shaped, so they swim by undulating their body. You don't see any, it doesn't look like a fish, and you don't see any fins. Okay, and again, no major bones. This is what the fossil looked like. And if you looked into the anatomy, okay, and by the way, this number here, 2.3, this is the figure in the textbook. Okay, but you see that it did have, the brain, the notochord, the tail, so the things that make the gill slits to make a vertebrate are here. Okay, and notice it had a head or a head region, no gills, so it's the beginning of the vertebrate, what they call the vertebrate plan. Here's another one, and this is the fossil, Metasprigina. Again, this is a primitive chordate, and again, it's the transition between this primitive form into something that looks like a fish. Okay? And when we think of, or when we look at Metasprigina, you see that it did have a notochord. It didn't have fins. Did not have a well-developed head or cranium. It had these pharyngeal bars. This will be important when we talk about the pharyngeal bars are basically what supports the throat. Okay. Like all the other fishes at this time, was a filter feeder. So it just swam with the mouth open and things just went in the mouth. And it swam with this side-to-side motion. Then we start getting into more fish-looking vertebrates. And we have these fish that look like they're covered with armor. Okay? This is what the ostracoderms are. Again, we're still in the Ordovician, Devonian. Now, these fish didn't have fins like the modern day fish, so they were probably not good swimmers. Okay, this is what they look like. Okay, so ostracoderm means shell skinned. Okay, right here means shell skinned. Okay, because it looks like they're covered with this heavy armor. Okay. See here that like in this one on top, it consists of these large plates that just are overlapping. While this one here, the C, looks like they're smaller type of plates. Okay. So again, heavy armor. The mouth was fixed. So again, no jaw, just an opening. And they believed that they were filter feeders, so they just swam and um, filter food in their throat. Okay, they went extinct, and it was believed that one of the reasons was that they were outcompeted by the next set of vertebrates that had jaws. Here's some of the different type of strichoderms that existed, many different shapes. So let's go through some examples. Okay. Here are the Jurassids. Okay. So these are from the freshwater, again, Silurian Devonian. And I'm going to keep repeating that, but just something that you know the fish were in the Silurian Devonian. Okay. And when you look at this, you see that the head shield has these bigger plates, and then behind the head, the rest of the body has these smaller ones. Um, this would be ideal and advantageous because think about it, the body has to swim, like the body has to undulate, from side to side. So if you have big plates, it would be difficult for the fish to move. But with these smaller ones in the body, it's like a little chain suit. It can move from side to side, while the head can have the bigger shields for protection. Okay. Now, these were about less than a foot long, uh, and oops, sorry. One of the new concepts that I want to introduce is the type of tail or caudal fin. And I need to turn the camera on for that. So hold on 1 second. So when you think of, when you look at that picture, you see that the tail, the actual bones of the tail, hold on, are going into the let me point to it, hold on, are going into the bottom lobe. Like if this is the caudal tail, and has the dorsal and the ventral, it has the bottom lobe. Let me turn this on. Oh, hold on, I gotta stop sharing. Hold on. So now, okay, so let me share this. That. Okay. So when you think of a fish, okay, think of like imagine that's a fish. Okay, happy fish. And there's the tail or caudal fin. So the modern day fish, if you looked at this part right here, okay, so that's the end of the body. The spinal cord comes down to here, and then there's a little fleshy part, but the bones of the spinal cord don't go into the actual tail like that. In these ancient fish, the spinal column continues. So here's the spinal column, continues. Here's the fish, and it goes into the fin itself, the caudal fin. So in some cases, have this where the spinal cord goes into the bottom portion of the caudal fin. Okay. Now this is called hypocercal or the most common name is reverse hyper circle tail. And then the other one, the most common one, would be where this just continues, and then you have something like, ooh, that was bad, like that. Okay, that's really bad. Let me do it here. So here's the fish, here's the body, here are the bones coming, and then they continue. This is the bottom lobe of the tail, and then this is the upper lobe of the tail, so like that. Okay, so this is the hyper circle tail. Okay, and this is what you find in sharks, modern sharks. Now, this is the most common. It's called hypercircle. This is the less common, and it's called hypocircle, but the majority of biologists just say the reverse hypercircle, the reverse of that one there. Okay. So I'm going to be pointing these things out to the to the different fish that we're going to be studying. So let me go back to the PowerPoint. Okay, so we're talking about the this. You see that the tail is reverse heterocircle tail. And again, this is when you see the hyper and the hypo is an indication of an ancient, in terms of evolution, ancient fish. Okay. And present day, the sharks and lampreys, they still have this. Another example of a Stracoderm are the order Anaspids, and the species will be Jamoidus. Now this one looks a little bit more fishy, but you still don't have the normal pair of fins that you find on the modern fish. Okay. Again, they were freshwater, present in the Silurian and Devonian, and then they went extinct. They were about 1 foot long, and almost no skeleton. See here by the picture, and also there, that it is a reverse heterocircle tail or caudal fin, and you can, like the other fish, the mouth is pretty much at the end, at the terminal end of the body, and you can see them swimming. You can imagine them swimming with their mouths open, and then just filter feeding the food in the water. This one had more fins. You see, they had it has a dorsal and a ventral fin here, so it's a little bit more elaborate, and it also looks a little like a common day a lamprey and hagfish, but it's not. It's not, as far as we know, not a relative or an ancestor of them. Okay. Okay, so now in this group, we have the cephalospeudomorphs. Again, they're still ostracoderm fish. They still have the no jaw, and these are sometimes more, what should I say, more like the different types of shape that you find in ostracoderms. It's a greater variety in cephalospeudomorphs than to the other ones, than in the other groups that I just mentioned. Okay. Now they have this big head shield that you see here, okay. And it looks like they have fins on the side of the head, but they don't. This is actually just part of the head shield. Okay, the eyes are on the dorsal side. Okay. You see that the tail is heterocircle, okay. And then ventral, the bottom of the body is flat. And what this indicates is that these fish were most likely bottom dwellers. They were bottom fish. So they would probably on the bottom of the ocean or lakes and just swim along. And when they found something dead or some food, they would just suck it up into their mouths. Now, the mouths had an opening, but again, not a jaw. So what they had is they had these small plates that made, if you think of like lips that were flexible enough to bring food inside. And this is what I was referring to when the people looked at the anatomy of that head shield. They saw that it had a series of grooves all throughout the different types of head shields, and this led them to believe that there was some type of sense organ, perhaps sensing food or being able to detect food in like a cloudy bottom, murky water environment. So then we go into the cyclostomes, the living agnathans, the lamprey and the hagfish. Now, cyclostome means circular mouth, Okay, and you see why. Here's the mouth of a lamprey. So again, no true jaws yet, just an opening. So these have no paired fins like a modern day fish. Their skeleton is cartilage. They still have the notochord, and then they have the gills in these pouches that have these little openings to the outside. Now, when you look at the taxonomy, again, this is from the textbook. You see that they're here on the left side. So you have the early vertebrates, and you have the hagfish and the lamprey. And you look at the connection and question marks, means that we don't really know how the two are related. We don't know how they, what they came from, what they are connected to. We don't know a lot about them. The two that living today are the Mycene, which are the hagfish, and then the Petromyzontidae, which are the lamprey. Okay, so here's some pictures of lampreys. And in lab, we will be we do dissect a lamprey or a hagfish. Okay, and it's usually a lamprey. See that the mouth consists of these rows and rows of little teeth. So basically, they grab onto a host like this fish. These teeth attach themselves so that it's not just suction, it's that the teeth actually hold on to the prey. And then they have a, here's another picture. Oh, here's another picture. Close up. Another one. Okay. So here he shows you on the top drawing, the mouth attached to the skin of a fish, and then this tongue that has this rasping structure, okay, comes down and cuts cuts the flesh and starts getting into the body, and then obviously the fish will start bleeding. The lampreys secrete this anticoagulant liquid, so that the fish just keeps bleeding, and basically they just feed off the liquid of the contents of the body of that fish or that host. Okay? Kind of like a horrible way to die if you think about it. Something just sucking your life, and you're still alive. Okay. The life history of lamprey is simple. They live in large freshwater lakes or rivers or the ocean. So they're found in different environments. in the adults, the mature adults, make these migrations up to small streams in the spring. Um, both sexes build this nest made up of rocks, and they move them, the little stones, with their mouths. They spawn in big groups, large numbers of eggs are deposited, and then they die. Okay? Just like you think of like salmon. Okay? Now the larvae, they call Ammocetes, they burrow in the mud of that stream, and then they are filter feeders for about 4 to 7 years. And then after they go through a metamorphosis and look like a tiny lamprey, then they start going downstream to the lake or ocean. This is to show you what the Ammocetes larvae look like. And the link is just for, just to show you, it's a PSA showing you what harm these larvae can do, especially in the Great Lakes. So the other one, the other agnathan, the living agnathan, is the hagfish. Now they look like the lamprey, but they're very different. Again, there's between 3 and 11 genera. These are only found in the ocean. They are strictly marine. They are also temperate and cold water, up to the subarctic. They live in deep, as in like half a mile deep water, high salinities, muddy bottoms. Okay. They're different animals. And one of the interesting things about them is, for a long time, they thought that they would only find one or the other sex. It's just showing them in a bucket, and the eggs, they produce large eggs. But they thought these were protandros, which means, let me go back here so I can type it. Okay, so the hagfish were believed to be pro, and then tandros. Okay, protandros, and what this means is that they were first males, and then they changed sex and became females. So this is something we'll be talking about, but there are fish that are protandrous hermaphrodites, so they're born one sex and then they change sex. Okay, but what really was going on is that the hagfish are born with an ovotestis, so they're born with undeveloped ovary and undeveloped testes, and only one of them develops. So either the testes or the ovary develop, the other one degenerates. So they're not really protandrous hermaphrodites like the other fish that we'll talk about are. So the mouths are not, if you look here, you don't see all the little rows and rows of teeth like the lamprey has. So what they do is to get it, so that they're not really parasitic, they're more scavengers. So when they find something dead, they attach themselves to the body, and then to rip it open, look on the bottom right here, they tie themselves into a knot. Okay, and then they move the knot closer up against the body. So this will be the fish here. And then by putting pressure against the body of the fish while holding on to the skin, they're going to rip it open. Okay. And then when they do, they get in the body of these animals and eat them from the inside out. So they eat, dead, disabled fish. They use their the tongue and their body to get into it. Again, they must, they're deep water, so they must have a good sense of smell to be able to find these things. And just think of another, you're getting, think of like a whale that dies and sinks to the bottom of the ocean. And then these hackfish find it, they open the body, get in it, but the inside of the body is not, it's not a welcoming environment. There's different liquids there, different osmotic, equilibriums going on, acids and bases and decomposing things. So they have to be able to adjust to that environment. Okay, and they do that by first they can. They're the only ones, as far as we know, that can have the osmotic equilibrium. So the inside of their bodies is at equilibrium with the outside. Now, this is common in invertebrates, but not invertebrates. Okay. But then they also produce this incredible amount of slime and produce it really fast. Okay. I left these links for you um, if you want to see a hagfish sliming a car in a couple of seconds. Um, but this is pointing to where the slime glands are. And basically another name for them are slime eels or snot eels, is that they can, they can fill a bucket of the slime in just seconds. And then the last group of the strachoderms are the conodonts. Remember, these were really, really common, but I said we don't know a lot of it about them. So basically, the name means cone tooth. So cone and then dont, tooth. And again, the extinct group, their taxonomy is very questionable. They, for many, many years, all that was known were just the teeth. This is all that was known until finally someone they found a body, found the imprint of a body, so they knew they looked like these eels with these giant or large eyes. They are considered index fossils. Remember I mentioned the index fossils. We know they are from the late Cambrian to the Triassic. So if you find a conodont fossil, you are in the Paleozoic time. Okay. Yes, again, so only for many years they were only known as from their teeth until they found some fossils in England. Okay. They are believed to be, or it is believed that they were filter feeders, not as complex as the other the ostracoderms, but they had the notochord, dorsal cord, tail, so they were vertebrates. Um, and they appear to be more developed than the lamprey and hagfish, so that they're not the ancestors of the lamprey and the hagfish. Okay, so that is the end for the first slide. Let me stop sharing, okay. And I'm going to stop the recording right now and then start a second recording.